So moving on, our next speaker is Dr. Gerry Zambetti from St. Jude Children's Research Hospital, who will be talking about the most common germline p53 mutation, R33H, is hypomorphic and associated with low tumor penetrance. Thank you.
I hope there's not a trapdoor up here. I want to begin by thanking the organizers for this opportunity to discuss some of the work that we've been doing in my lab at St. Jude Children's Hospital. The project really begins with our focus on p53 and the signaling pathway that it controls and how it relates to childhood cancers, and in particular, a rare form of childhood cancer called adrenal cortical carcinoma. So we've been privileged to work with Dr. Raul Ribeiro at St. Jude, who's originally from Brazil. Raul recognized that the incidence of childhood adrenal cancer is extremely high in Brazil, and so it's turned out to be really a fascinating project. So the project begins in a city called Curitiba, which is south of Sao Paulo, and the incidence of childhood adrenal cancer is about 15 times higher there than elsewhere in the world. And just a little bit of background about adrenal cortical carcinoma. It arises in the adrenal glands, which sit on top of the kidneys. And the adrenal cortex is involved in producing a variety of hormones that regulate blood pressure, glucose metabolism, immunity, sex development. And when this cortex becomes malignant, it's usually, in children, it's usually hyperactive, where it's producing large amounts of these hormones. And that's manifested by these phenotypes where there's virilization, where the child will develop facial hair. They'll develop pubic hair, body odor, acne. They've gone through puberty in every way. They'll have muscle fatigue. They'll have high blood pressure. They'll have palpable tumors, sometimes quite large in size. And so when the children come to the clinic with adrenal cancer, they're usually between 3 and 4 years of age. It's more common in girls than in boys. There's really no effective proven chemotherapy. They get a very aggressive form of chemotherapy. Really, surgery is the most effective option. If they can completely remove the tumor, prognosis is good. But overall, the survival rate's only 50%, and that number really hasn't changed very much in the last couple of decades. So usually when a child has adrenal cancer, it's usually within the context of Li-Fraumeni syndrome. And so here's a pedigree that's representative of a typical Li-Fraumeni family that's inherited a DNA binding mutation that's completely inactive, and that you can see that everyone who's a carrier of this mutation has gone on to develop some form of cancer in their life. And at the very bottom of the pedigree, you see that there's a child that developed adrenal cortical carcinoma. So it's usually seen within the context of Li-Fraumeni syndrome. But what we found was that the Brazilian families where there's children with adrenal cancer, there's no other instances of cancer within those families. You can't characterize them as being Li-Fraumeni-like. And this particular pedigree is typical. You have a mom who had a child with one father. That child developed adrenal cortical carcinoma and died. She remarries and has another child. That child developed adrenal cortical carcinoma. So one mom and two children had adrenal cancer. The normal instance of adrenal cancer in the general population is 1 in 3 million. So you can see that something's unusual in this circumstance. And what we came to learn is that these children in Brazil have inherited a p53 mutation. And it's not your standard Li-Fraumeni DNA binding mutation. It's in the C-terminus, in the dimerization, right out of salt bridge, where the histidine substitution would impact this dimerization domain and the stability of the protein. So to summarize a large amount of work, what we found that almost every child from southern Brazil have inherited this 337 mutation, even though they come from different families. And what we've come to learn from work by Amelia Pinto and then Maria Isabel Oschatz and Pierre Hanot, this mutation is a founder mutation. It began on a Caucasian allele in Europe, probably more than 2,000 years ago, and maybe several hundred years ago, it was brought over to Brazil. We did a lot of work on this particular mutation to see what's the consequence on P53 function. So we generated a cDNA, we reintroduced it into a variety of different cell lines and tested it in many different P53 assays. And in every case, it behaved just like wild-type P53, not like a DNA-binding Li-Fraumeni mutant. So it's different. It retains its activity. And perhaps that's why in the families that we looked at initially, there wasn't massive cancer incidence. We worked with Richard Krawacki at St. Jude. He's a structural biologist, and we showed that this mutation has a consequence on the structure of P53, and that it affects the structure in a pH-dependent way. And when we looked at the tumors were telling us that there was a problem with this mutation. Always, they've undergone LOH where they've deleted the wild-type allele, they selected for the mutant allele, and the missense proteins expressed at very high levels in the nucleus. If it was doing its job, it should be blocking the growth of those adrenal tumors or killing those adrenal tumor cells. So clearly, it's defective in that physiologic setting. The other compelling evidence that it was related to these adrenal tumors is that, excuse me, one in 10 carriers go on to develop adrenal cancer. And again, the normal incidence is 1 in 3 million. And perhaps the adrenal tumors arise in these carriers, perhaps due to the physiology of the adrenal gland. And we can talk more about that after the presentation. So bottom line is that the majority of the families that we initially studied, there were 30 families. Most of those families, 23 families, had no family history of cancer, but 7 families did. There was occasional breast cancers, brain cancers, tumors that you would see in a Li-Fraumeni-like setting. So this suggested that there was families with a founder mutation that were at low risk for cancer and other families with the identical P53 mutation at high risk. And that was verified and extended by Maria Isabel and Pierre, where they screened for patients that fulfilled the criteria for Li-Fraumeni-like in southern Brazil. And they found that there were individuals that met that criteria that had the 337 mutation. So these would be representatives of high-risk cancer families. We worked with Juliana Asumco, where we were able to access a group of breast cancers in young women from southern Brazil, and it was, we found that germline 337 was associated with those cancer cases as well. And what we also found in that study is that there are polymorphisms within P53 and in the MDM2 promoter. These are polymorphisms that are expected to further reduce P53 tumor suppressor activity. And we think that it's the combination of the 337 mutation plus these polymorphisms that raise the risk of those carriers to develop breast cancer. But I want you to keep in mind that all of those studies had a bias built into them. Our initial study where we screened for children with adrenal cancer, there had to have been a child that developed adrenal cancer to enter our study. For Maria, Isabel, and Pierre, there had to be an individual that met the criteria for Li-Fraumeni-like. In this particular case and in other breast cancer studies, there had to have been a woman who developed breast cancer to enter that study. And so that can introduce some biases that could influence the outcome. And so to try to avoid those filters, we were able to do a general population screen using a very simple PCR test. And it was done in collaboration with a colleague of ours, Bonald Figueiredo from Curitiba, Brazil. Bonald screened 175,000 newborns. And remarkably, 1 in 375 have inherited this germline mutation. And although this study was done in Curitiba in the state of Paraná, similar findings were made in Sao Paulo and Rio de Janeiro. It's a total population of 80 to 100 million people, and we estimate that there's anywhere from 200 to 300,000 carriers of this germline p53 mutation. What we also were able to do without the filter, we found that 1 in 40 of the carriers went on to develop adrenal cancer. So not quite as high as our initial prediction of 1 in 10. Again, probably related to the bias of having that child having developed adrenal cancer to enter the study. So 1 in 40 is still significantly higher than 1 in 3 million. But it also means 39 out of 40 don't develop adrenal cancer. There was some clinical benefit to the screening where families were invited to bring their child back to the hospital every few months for a simple blood test. Any change in adrenal hormones, they would follow up with imaging, and they were able to detect tumors at early stage where they were easily resected, and outcome is good. Families that didn't agree to the follow-up did come back with their children with later stage disease, and there's been several deaths as well. And so there's a clinical benefit, at least within this setting where there's such a high incidence of this germline mutation and high risk for adrenal cancer. And this general population screen confirmed the original set of data where there's low risk carriers and high risk carriers. How can that be? And so we're trying to understand how this mutation affects p53 function and its tumor suppressor activity in a physiologic way. And so we were able to generate the knock-in mutant mouse. So we introduced this single point mutation in the germline of mice and then tested the activity of that mutant in the mouse, in the animal model. These mice are born with near Mendelian frequency. There's a slight reduction in homozygous mutant mice. There's a reduction in female mice, but it's not statistically significant. The first experiment that we did was to test the function of this endogenous 337 mouse protein in thymocytes in response to DNA damage. So this is following Scott Lowe's initial apoptotic assay. And what we find is that wild-type thymocytes, untreated, are viable. And this is done in short-term cultures. So we take the thymus, we get isolated thymocytes, half are untreated, half are treated with ionizing radiation. We then stain those cells with propydium iodide and then FACS sort them. And so the wild-type thymocytes are fine when they're untreated, and then after DNA damage, they completely lose viability. They've undergone a massive apoptotic response. The DNA becomes fragmented, and that's shown by this sub-G1 peak. p53 knockout thymocytes are completely resistant to DNA damage, and that's what's shown here in the bottom panel. And then in the middle panel on the left side, you can see the response of the homozygous mutant mice. And it's in between. Clearly, they survive better than the wild-type thymocytes, but not as well as the p53 knockouts. So it's consistent with it being partially active, hypomorphic. And in the upper right panel is a Western blot showing that the 337 mutant protein is grossly overexpressed in the thymus, even when it's untreated. And that's compared to the wild-type mice, wild-type thymocytes, and to p53 knockouts. And then if you go down to panel C, the middle panel of Western blots, and you look at the middle panel of that Western blot, you'll see in the thymus. So these cells are not in culture. This is right out of the animal, snap frozen. You can see that the 337 mutant is grossly overexpressed in the thymus, untreated animals, much higher than wild-type p53 after DNA damage, and much higher than the 175 mutant, DNA-binding mutant that would be associated with Li-Fraumeni. It's different than that particular mutant. And then in the bottom set of panels, in the red in the second line, you can see by immunohistochemistry that the 337 mutant protein is just naturally, abnormally expressed in untreated thymus. So it's abnormally expressed, and it's hypomorphic in response to DNA damage in thymocytes. We then challenged it in a model of Burkitt lymphoma, where it's an emu-MYC transgenic animal, where the MYC oncogene is overexpressed in B cells. These mice get B-cell lymphoma, usually by around 4 months of age. And work done by Christine Eischen in John Cleveland's lab in collaboration with Chuck Scher and Martine Roussel, they showed that when you overexpress MYC in lymphocytes, they undergo apoptosis. And for these B cells to become lymphoma cells, they have to select against the p53 pathway, either by deleting ARF, by mutating or deleting p53, or by overexpressing its negative regulator MDM2. And so we crossed the 337 mouse into this emu-MYC transgenic, and what we found was that the homozygous mutant mice develop B-cell lymphoma with exactly the same latency as the wild-type mice. So if 337 was significantly compromised, these mice would be developing B-cell lymphoma either at birth or shortly after birth. And then we also showed by analysis of the B-cell lymphomas that do develop in these homozygous mice that they end up selecting against 337 by introducing second site mutations, either in the DNA binding domain or insertions or so forth, selection against ARF. So it behaves just like wild-type p53. The same mindset applies to primary MEFs grown in culture. They undergo senescence, and then rare cells grow out of those senescent cultures where they become immortalized. Those immortalized cells always select against the p53 signaling pathway. And so we challenged the ability of 337 to function within the senescence program. And what we found is that wild-type, heterozygote, and homozygous mutant MEFs all underwent senescence with the same kind of kinetics. And for those immortalized MEFs that came out of the homozygous mutant cultures, they select against that mutant. And in fact, one of the mutants that we identified is an alanine to 135 valine mutation in the 337 mutant allele. This is a TS allele that's been a very important reagent for the p53 field for some time. The other thing that we found was that 337 mutant protein, just as we saw in vivo, is abnormally high in the primary MEFs compared to wild-type p53. But despite its abnormal expression, it was still competent for upregulating p21, which is consistent with it undergoing senescence. And so those are a bit artificial models, but standard models for the p53 field. This is just looking at spontaneous tumors. And what we find is that the homozygous mutant mice in the red bar on the left panel do not fare as well as the wild-type mice, and that they are prone to cancer, but with low penetrance. Certainly in contrast to what we see with a typical DNA binding mutant shown in the right panel. And this is work done by Gigi Lozano's group. Mice that are homozygous for the 175 mutation all develop cancer between 3 and 6 months of age. As you can see from the Kaplan-Meier curve on the left, the homozygous 337 mice don't develop cancer within the first year. They start to develop some tumors in the second year. And those tumors include a fair number of carcinomas, a single case of sarcoma, and a number of lymphomas as well. We also see some non-malignant pathologies that caused us to have to sacrifice these animals, and these seem to be related to inflammation. And so there's some mice that develop nephritis, some sepsis, and other forms of inflammation. So just to summarize this part, the 337 mice represent the most common inherited germline p53 mutation. It's hypomorphic, where it has partial activity in thymocytes in response to DNA damage. It's abnormally expressed in vivo, even in unstressed tissue. It's abnormally expressed in MEFs, but still competent for enforcing senescence. Selected against in Bcell lymphomas and in immortalized MEFs. And so what we suggest is that 337 may have a consequence dependent on tissue type and stress response. We're challenging that hypothesis currently. We're also looking for changes in gene expression by Affymatrix profiling approaches. And we're also crossing 337 to other tumor models to see whether it can cooperate with other genetic lesions in regulating cancer susceptibility. And so what we've shown is that 337 clearly is not as competent as wild-type p53, but clearly not as incapacitated as a DNA-binding Li-Fraumeni mutant. Hence the concept of it being hypermorphic and an in-between type of mutation. And those findings suggest that for those families where they do have a high risk and have a phenotype that would be LFS or LFS-like, we suggest that there's a cooperating genetic alteration. And so we're trying to understand what that cooperating genetic alteration may be. So I just want to present some very preliminary data that we've recently generated. And it was derived from our studies doing whole genome sequencing. So we published on this not too long ago. Amelia Pinto will talk more about it later on in the meeting. But we've been able to do whole genome sequencing on a cohort of Brazilian adrenal cortical tumors. And what we found was that the WGS approach, whole genome sequencing analysis, identified a second germline mutation in a putative tumor suppressor in a subset of those adrenal tumors. And what we've been able to do is to access a fair number of Li-Fraumeni associated tumor types from Brazil, including breast cancers and sarcomas. And they're from 73 independent families. So each tumor is an independent family, representing an independent family. And they're all 337 positive. And what we found was that 90% of them have both mutations within this putative tumor suppressor plus 337. And so we believe that this second site mutation is cooperating with 337 to raise the risk, hopefully explaining why there are low-risk carriers, high-risk carriers. And we're challenging that hypothesis by introducing that second site mutation into our 337 mouse. I just want to make one comment about this slide. I call it the gradient effect. Not every mutation is equivalent in p53. And I want to emphasize the point that there could be a mutation at codon 175. It's almost always a histidine mutation. That histidine mutation results in a zinc finger that causes the protein to misfold. With it misfolded, it's completely inactive. We have adrenal cases, an adrenal case at St. Jude, where it's a 175 leucine, germline mutation. That leucine can't form the zinc finger, so it can maintain its conformation, retain significant amount of activity. And for those carriers, their susceptibility to cancer is much lower than if it was a 175 histidine mutation. So I just wanted to introduce that concept in this forum. And I'm going to stop there and just acknowledge the help of our collaborators. Thank you very much. Okay. Hi, Carol.
Hi. The levels of P53 in the MEFs are so high compared to wild type. Have you not looked at MDM2? Perhaps this is defective in inducing MDM2, or is it at the level of RNA of this?
It's defective in inducing P21 and MDM2 in response to DNA damage.
But even the basals seem very, very high.
Correct.
Did you look at MDM2 under those conditions?
So we have some data to say that that's the case, that MDM2 expression is diminished. We're also doing some biochemical analysis where we're looking to see whether it forms tetramers in vivo. Obviously, some of those approaches are more technically challenging. But the prediction is that it's compromising its ability to form a stable tetramer. If it can't form a stable tetramer, it's not going to be upregulating MDM2, and it shouldn't be recognized by MDM2 and not turned over. And that would be consistent with its stable half-life. So we're trying to sort those experiments out.
Gerry, terrific work. Is anything happening in the adrenal cortex or the adrenal gland in these mice? I mean, what is the sort of similarity or differences between the two, but humans and mice? And is there actually something subtle going on there that we don't know yet?
Yeah, so that's a really good question. So it turns out that the mouse adrenal physiology is different than human. And so it may not be the ideal model for us to study the adrenal cortical tumors, but I think it's still a valid model for studying all the other types of cancers that we can see. With that said, we had a heterozygote mouse that developed an adrenal cortical carcinoma, which is pretty rare in the mouse model. So that would be at least consistent with some of the carriers we see in Brazil. And I'll talk to you more about the physiology of the mouse adrenal later. Okay. Thank you.